Glycogen phosphorylase, liver form, PYGL, also known as human liver glycogen phosphorylase, HLGP, is an enzyme that in humans is encoded by the PYGL gene on chromosome 14. This gene encodes a homodimeric protein that catalyzes the cleavage of alpha-14 glucosidic bonds to release glucose 1-phosphate from liver glycogen stores. This protein switches from inactive phosphorylase B to active phosphorylase A by phosphorylation of serine residue 14. Activity of this enzyme is further regulated by multiple allosteric effectors and hormonal controls. Humans have 3 glycogen phosphorylase genes that encode distinct isozymes that are primarily expressed in liver, brain and muscle, respectively. The liver isozyme serves the glycemic demands of the body in general, while the brain and muscle isozymes supply just those tissues. In glycogen storage disease type 6, also known as HERS disease, mutations in liver glycogen phosphorylase inhibit the conversion of glycogen to glucose and results in moderate hypoglycemia, mild ketosis, growth retardation and a hepatomegaly. Alternative splicing results in multiple transcript variants encoding different isoforms. Provided by RefSeq. February 2011. Structure. The PYGL gene encodes one of 3 major glycogen phosphorylase isoforms, which are distinguished by their different structures and subcellular localizations. Brain, PYGB, muscle, PYGM, and liver, PYGL. PYGL spans 846 amino acids and shares fairly high homology in amino acid sequence with the other 2 isozymes, with 73% similarity with PYGM and 74% similarity with PYGB. Nonetheless, PYGB and PYGM demonstrate greater homology to each other, indicating that PYGL evolved by a more distant descent from the common ancestral gene. This protein forms a homodimer, with each monomer composed of N-terminal and C-terminal domains of nearly equal size. The catalytic site forms at the interface between these 2 domains and interacts with the required cofactor, pyridoxal phosphate, to bind the substrate glycogen. This cofactor is attached by a covalent Schiff-base linkage to Lys 680 in the C-terminal domain. At the opposite side of the enzyme, the regulatory face opens up to the cytosol and contains the phosphorylation peptide, which is phosphorylated by phosphorylase kinase and dephosphorylated by the phosphatase PP1, and the AMP site, which is connected to the active site by an adenine loop. Phosphorylation, a binding of the allosteric sites, induce conformational change that activates the enzyme function. As a glycogen phosphorylase, PYGL catalyzes the phosphorylasis of an alpha-1,4 glycosidic bond in glycogen to yield glucose 1-phosphate. Degradation of glycogen, the glucose 1-phosphate products, then contributes to glycolysis and other biosynthetic functions for energy metabolism. As the major isozyme in liver, PYGL is responsible for maintaining blood glucose homeostasis by regulating the release of glucose 1-phosphate from liver glycogen stores. One model suggests that circa 2+ oscillations play a role in activating glycogen phosphorylase in glycogen degradation in liver cells. Through its function in the liver, PYGL is also central to meeting the glycemic demands of the entire body, though other tissues may express all 3 forms in different proportions. The purpose of expressing multiple glycogen phosphorylases remains unclear. Clinical significance. PYGL has been implicated in glycogen storage disease type 6, also known as HERS disease, and both type 1 and type 2 diabetes. Glycogen storage disease type 6 has been attributed to PYGL deficiency as a result of causal mutations in PYGL gene, including 2 splice site mutations and 2 missense mutations. The function of PYGL in regulating liver glucose production also plays a role in diabetes, since hypoglycemia in type 2 diabetes is the result of excessive glucose production by the liver. Developing a drug that targets PYGL may prove effective in controlling blood glucose levels. Glycogen-induced hepatomegaly in type 1 diabetes and glycogen storage disease type 6 present similar clinical manifestations such as liver dysfunction, fasting hypoglycemia, and ketosis. Interactions. Inhibitors. PYGL has been known to interact with allosteric inhibitors, including BioW1807 and sugar derivatives that bind the glucose inhibitor site. In addition, glucose and purines stabilize the inactive conformation of PYGL, thus inhibiting binding to its active site. See also glycogen phosphorylase, PYGB.